Hello, everybody. Welcome to the narration of Chapter 14, Part 1. Um, we have seen in this class the basic workings of the nervous system in Chapter 12. We put most of our emphasis on the workings of the neurons. And in Chapter 13, in the spinal cord chapter, we have seen some actual wiring diagrams of how neurons course through the body, especially through the spinal cord, and how they are connected to create those reflex arcs, those automated responses to changes in the environment. Well, in the case of the brain, the reflex arcs are there, but the wiring diagrams are a lot more complex. So we will not revisit or we will not explain any detailed wiring diagrams, but we will mention quite a few of these reflex arcs. We may call them by different names, but whenever I can, I'll remind you that we are talking about a reflex arc, an automated response to changes in the environment. In terms of the gross anatomy of the brain, and that will be our guide throughout this chapter, when you look at the sagittal section of the human brain, even to the naked eye, there are four very different looking parts showing up. Here's this largest wrinkled looking portion of our brain, the cerebrum. We have this much smaller and grooved or dotted surface area. This is the cerebellum. We have the continuation of the spinal cord, which ends about here. This straight, relatively featureless portion of our brain is the brain stem. And then right here, we have something that is called a diencephalon. This is the last fourth part of the brain. We're going to spend time here, and surprisingly, this portion of our brain that will keep coming up in its functional diversity in many organ systems in AMP2. So when you look at the artist's rendition, you are seeing the same four parts, except the diencephalon is a little bit skewed. One portion of the thalamus is maybe a little overemphasized, but nonetheless, what we're going to do in this chapter is looking at gross anatomical structures, sometime on the surface, sometime deeper in the brain, and connect them with function. And that function is usually going to be part of a reflex arc, part of an automated response. So in this first part, we're going to start with the brainstem, because that connects us to the previous chapter, the spinal cord. And then we're going to take a look at the cerebellum and the diencephalon. In the second part, we're going to talk about the cerebrum and the cranial nerves. Few general anatomical or metabolic facts about the brain. Our brain uses tremendous amount of oxygen and glucose. You saw that in the muscle chapter that to create ATP, we need an energy carrier, glucose, which will be degraded through anaerobic and aerobic respiration. Aerobic respiration needs oxygen. That kind of chemical reactions go on in our mitochondria. And so any organ that needs a lot of ATP will need a good deal of oxygen and glucose or some other energy carrier. Why is our brain demanding so much oxygen and glucose? Remember that resting membrane potential from chapter 12? to set resting membrane potential, just to provide the basic starting condition for that 100 billion neurons in our brain, we need that sodium-potassium pump. That's an ATP gobbling little molecular machine, and that is why we need so much oxygen and glucose for our brain, because we have to keep those pumps going 24/7. With every action potential, we have to reset the resting membrane potential in that neuron. Think about 100 billion neurons. Our brain works 24/7, even if when we are asleep. The non-consciously working portion of our brain, like brainstem and especially diencephalon, keep on adjusting homeostasis in our body even when we are no longer conscious, like when we are asleep. So nerve impulse firing goes on 24/7 in our brain. It demands, that fact demands a lot of oxygen and glucose. Why glucose? On the next slide, we'll explain that many organs in our body can use fatty acids to power mitochondria. Our brain cannot. Combine that with the fact that storing carbohydrate in the brain is limited. For some reason, our neurons cannot store glycogen, and so our brain is relying on the blood bringing continuous stream of glucose to it. Our blood sugar level is set at 700 milligrams per deciliter glucose. Hypoglycemia, when our blood sugar levels go below normal, affects our brain most decisively, and so our brain starts to malfunction, especially the consciously working part of our brain, and confusion, dizziness, sometimes convulsion, but fainting occurs when the brain doesn't get enough glucose. And the same symptoms occur when we don't have enough oxygen. Think about somebody's passing out because of choking, because of any obstruction in the airways, where our respiratory system can no longer bring oxygen to the blood. Oxygen and glucose are mandatory requirements for continuous working of the brain. Why the brain cannot use fatty acids for energy production? The answer is right here. This also takes us back to that warm and fuzzy corner of chapter 12, the neuroglia. Astrocytes, part of the neuroglia, form the blood-brain barrier for most of the brain. The last slide in this lecture will be about some parts of the brain that are missing the blood-brain barrier. Those are the curiosity, the unusual part of the brain. So in most areas of the brain, fatty acids and some antibiotics, some chemotherapy drugs against brain cancers are also prevented entry into the brain tissue. So it's not only fatty acid, but many other small organic chemicals are refused entry by the blood-brain barrier. With the spinal cord, we mentioned all the details that you need to know of the meninges. Here you can see the meninges covering the spinal cord. Meninges form a continuous sheeting that comes through the foramen magnum and continues on to the brain. Gross anatomically speaking, the sheeting of the spinal cord is like a tube. When you look at the brain, in between brain portions or different parts of the brain, there are these huge invaginations of the meninges. And so we have up here, and we're going to see this up close in a few slides later, the faux cerebri that is reaching in between the two hemispheres of the cerebrum. We have the tentorium cerebelli, wonderfully named, this big finger-like invagination of that purple membrane. And we also have one in between the hemispheres of the cerebellum, the faux cerebelli. Just to review from chapter 13, the layers of the meninges are the dura, the outer layer. Deep to it is the arachnoid matter, and the deepest right on the surface of the brain and spinal cord is the pia matter. In the spinal cord chapter, we mentioned that between the dura and arachnoid there is tissue fluid. Between the arachnoid and the pia there is this very special cerebrospinal fluid. Tissue fluid just oozes in there. Cerebrospinal fluid between the arachnoid and pia needs to be made in a special portion of our nervous system. So those ependymal cells that make cerebrospinal fluid are located in the brain, and so we're going to spend a little more time on how cerebrospinal fluid is produced and drained from our central nervous system. When you look at cerebrospinal fluid, it's a yellowish, clear liquid. It has glucose, useful material for all neurons, and it has ions. And when we say ions in a nervous system, sodium, potassium, calcium ions are the ones that meaningfully impact, maintains the balance needed for action potential or synaptic transmission. So we do get a mechanical effect out of the cerebrospinal fluid. We get that from a tissue fluid between the dura and the arachnoid as well, providing a cushioning layer to make the impact with the bony protection a little less severe. And in the case of our brain, the bony protections are coming from those bones of the cranium. We looked at those in the bone lab and chapter 7, the exoskeleton. We receive a little bit of chemical protection, glucose and ions, sodium, potassium, calcium ions are in this fluid that neurons may use. We also have a circulation of the cerebrospinal fluid, and this just means that cerebrospinal fluid is made in the brain and also drained away there. A clinical analytical aspect of cerebrospinal fluid that this can be obtained through a fairly simple procedure, and spinal tap, the procedure to obtain cerebrospinal fluid, is carried out millions of times in clinics. What is cerebrospinal fluid good for? Because cerebrospinal fluid contains waste products from the brain and spinal cord. The gold standard diagnosis of meningitis. Is it viral? Is it bacterial? Well, those bits of bacteria or viruses will be swimming in the cerebrospinal fluid, so sampling and analyzing cerebrospinal fluid will help determining the type of meningitis or diagnosing meningitis. The gold standard diagnosis of dementias, is it Alzheimer's disease? Is it a Lewy body disease? Is it a Pick's disease? There are many forms of dementia besides Alzheimer's disease. Gold standard diagnosis still comes from cerebrospinal fluid. And we have several psychiatric disorders that can be diagnosed, or drug utilization can be analyzed from cerebrospinal fluid. So where cerebrospinal fluid is made, this introduces us to the concept that inside our brain there are large fluid-filled cavities called ventricles. And so this is the best view of the ventricles. Here are the two lateral ventricles. Here's one, this C-shaped sausage-looking structure is the one of the lateral ventricles. A little bit behind it, you can see the other side. We do have the third ventricle. This is about the size of a quarter. It's thin as a coin. And the fourth ventricle is right between the cerebellum and the brainstem. You can see that cerebrospinal fluid can flow in the middle of the spinal cord through the central canal. So the places where the cerebrospinal fluid is made, the choroid plexuses, are located inside these ventricles. Once the cerebrospinal fluid is made in those ventricles in the choroid plexus portions, we need to drain it away because accumulation of the cerebrospinal fluid would cause a swelling between the pia and the arachnoid matter. On this particular illustration, the pia is red. You can see how it follows every nook and cranny, the surface of the cerebrum. The arachnoid matter is purple, and you can see the space between the pia and arachnoid filled with cerebrospinal fluid. The dura is this light green, much thicker material. What happens with the arachnoid layer is that extensions of the arachnoid layer, arachnoid villi, poke through the dura and release surplus cerebrospinal fluid into venous blood. So you can see the formation and therefore the reabsorption rate. 20 milliliters an hour means that in a 24-hour period, our choroid plexuses make enough cerebrospinal fluid to fill a regular water bottle, plastic water bottle. So quite a bit of fluid is produced here, but again, production and drainage are imbalanced, so there is, under normal circumstances, there is no stretching or expansion of the arachnoids, subarachnoid space. So this is the best summary view of cerebrospinal fluid, and here you see individually named choroid plexuses. Choroid plexuses use blood and filter it in a way to create this excuse me, create this clear fluid, cerebrospinal fluid. So the lateral ventricles, third, fourth ventricle, they all have choroid plexuses. When you look at this image, the choroid plexus of the third ventricle is visible best. You can see the purple arrows excuse me. I always get emotional when I talk about choroid plexus. Sorry about that. So the choroid plexus of the third ventricle is where you see the beginning of these purple arrows. That indicates cerebrospinal fluid is made there. But in the fourth ventricle and in the lateral ventricle, this happens as well. This is also important. These ventricles are connected. So cerebrospinal fluid can flow from the lateral to the third, to the fourth, and all the way to central canal. You see that between the arachnoid and pia of the spinal cord, cerebrospinal fluid is circulating upward, and the drainage happens right here in this area through the arachnoid villi. And so we started from blood to make cerebrospinal fluid, and we are returning surplus cerebrospinal fluid into blood. And here begins our journey through the four parts of the brain. We are starting with the brain stem. And so let's list functions and connected structures. Because the medulla oblongata, the first part of the brainstem, is a connection, is a continuation of the spinal cord. Just like we saw the white matter aspect of the spinal cord, the ascending and descending tracts, they continue through the medulla oblongata. There's no abrupt stop to all those structures just because the spinal cord passed through the foramen magnum. So our central nervous system is one continuous structure. This foramen magnum just serves an artificial place to call one part brain, the other spinal cord. So there's no break here. The sensory and motor tracts continue. In terms of gray matter, coordinating centers for reflexes, our cardiovascular system, heart and blood vessels are kept functional with the help of the cardiovascular center. So this is actually contains several centers of our cardiovascular system, and so several reflex arcs have their integrating center here. We do have an integrating center for our respiratory system, medullary rhythmicity area, and we'll see this more in the respiratory center. We have a pacemaker for our inhalation. When we are sleeping, we still inhale 12-15 times a minute, and that is rhythmic activity is coordinated here. We also have reflex centers, coughing, sneezing, swallowing. These are also, I should add, vomiting, and some other centers related to the digestive system located here. Coughing and sneezing and even swallowing interrupts the regular activity of the respiratory center, and these are all automated. When we cough, no matter whether our coughing starts when we inhale or exhale, these reflex centers disrupt automatically the functioning of the pacemaker and allow us to cough, sneeze, swallow. So again, a number of automated reflexes are coordinated here in the medulla oblongata. We have connection to the cerebellum, but we'll see that later on a little better, especially with the midbrain and the pons. When we look at the little detailed dissected view of the medulla oblongata, the anterior view is showing us, maybe most visible is these bright yellow origins of cranial nerves. Cranial nerve 8 through 12 originate from the medulla oblongata, and that means that cranial nerve 8 should sound familiar. This is the auditory nerve or vestibulocochlear nerve. That cranial nerve had a passageway through the temporal bone, and we looked at that in the lab. Other structures, the white matter structures are the pyramids. These pyramids are carrying motor information out of the brain, going to the spinal cord, and eventually to skeletal muscles. We come back to this in chapter 16 in MP2, but if you take a closer look, there's a crossing of one bundle over the other, and this point called the decussation or crossing. So nerve impulses directing the left side of our skeletal muscles are coming from the right side of our brain, and nerve impulses that go onto the muscles on the right side of our body coming from the left side of our brain. In terms of gray matter, this is something interesting to mention. The olive is named because of its shape and size that is comparable to an olive. As you can imagine, this is a very ancient name, and when this was named, nobody had a clue what this olive did. It turns out to be a collection of the gray matter and signals of proprioception, how our body moves, how our balance changes, is switched over to new sets of neurons and sent to the cerebellum. The next part of the brainstem is the pons. The pons also has, just like the medulla oblongata, ascending and descending white matter tracts. We do have gray matter organizing, coordinating centers. We have the pneumotaxic, aneuploidic areas associated with adjusting respiratory rhythm and activity in our body. And here is yet another set of connections to the cerebellum. The cerebellar peduncles connect to the cerebellum, which is right here. Why is this connection from the medulla oblongata and from the pons? Our cerebellum is siphoning off a lot of sensory information coming from our proprioceptors. Our cerebellum coordinates skeletal muscle action, and it needs a lot of sensory information, knowing what muscles, limbs, entire body does in three-dimensional space. So a lot of information from these early parts of our brainstem streaming into the cerebellum. We also see from the pons, cranial nerves 5 through 7 originate. Midbrain is the last portion of our brainstem. It has a giant opening, the cerebral aqueduct. You can see a little better the choroid plexus of the third ventricle here, and here the choroid plexus of the lateral ventricle. And you can see the black arrows representing movement of cerebrospinal fluid. So cerebrospinal fluid has its own little duct that allows cerebrospinal fluid to go into the fourth ventricle right here. The midbrain has a little more, a few more gray matter areas. Let's talk about this substantia nigra, this dark band of neurons here in the midbrain. Substantia nigra and the red nucleus help maintain muscle coordination during movement and maintaining balance. Substantia nigra is one of the areas that is shrinking and damaged in Parkinson's disease. We also have to mention cerebral peduncles. Some nerve impulses and bundles of axons are going into the cerebrum. In addition, some others going into the cerebellum. And you also see here, this is the aqueduct. This is the opening for the cerebrospinal fluid going into the the fourth ventricle. A little more weird aspect of coordinating centers. Again, these are part of reflexes in a midbrain. If you take a little closer look at a dissected midbrain, you would see these bumps that were named colliculi. Each bump is a size, maybe a size of a black pepper or so. So they are visible to the naked eye, but they are not huge by any means. We have the superior set of bumps, superior colliculi. They coordinate the reflex. Whenever you look and turn to look at a bright, shining object or a sudden flash of light, our nervous system is programmed to react to that change in the environment, and this is where the coordinating center is. We are also programmed, hardwired to move our head into the direction of a loud sound, or if somebody is talking to us. So that reflex is coordinated by the inferior colliculi. And we have to mention cranial nerves 3 and 4 originate from midbrain. With relation to the brain stem, there is this unusual set of spotty collection of clumps of neurons scattered through the brain stem. Reticular activating system was the given name to this. So this is a group of neurons that alert and reboot when we are waking up the cerebrum. So when we go to sleep, the cerebrum portion of our brain is inactivated. We become unconscious. To wake up in the morning, something needs to tell this cerebrum portion of our brain, hey, it's time to get going. That signal is coming from the reticular activating system. This is programmed by the hypothalamus, and I'll say a little more about that when we get to the hypothalamus. It is the reticular activating system that allows us to have our sleep disrupted. Loud sound, bright flash of light, even somebody shaking our shoulders can wake us up. All those sensory inputs are coming into the reticular activating system, and this system wakes us up, reboots our cerebrum when we are awakened from our sleep. Our next large part of the brain is the cerebellum. There is a little more functional context coming to this in chapter 16 in HAPch14. The overarching function is to coordinate skeletal muscle contraction that maintains posture, a balanced position of the body, no matter what you're doing. So maybe the most outrageous example of regulating balance is in addition to learning to walk. Think about an infant not being able to do much movement right after birth, maybe uncoordinated movements of arms and legs, and then a year later, that little guy or gal running around causing all kind of mischief. So to get through that first year, the cerebellum constantly adjusting activity of those muscles of the legs, torso, and arms. So this body of this toddler can now maintain balance while standing up, and then start walking and running eventually. For later in childhood, this learning activity and adjusting activity of the cerebellum comes to play when we learn to ride a bicycle. Totally new ball game. New sets of muscles need to be activated as we push on the pedal, as we steer this bike and try not to fall. So anything we do, you learn a musical instrument, you decide to learn to play the piano. Guess what? Those fingers, their sequence, how they push the keyboard on the piano, need to be coordinated. You guessed it, cerebellum is the one who is learning how to coordinate your posture, movement of the arm, forearm, hands, and fingers for playing the piano. How does the cerebellum does that? Well, the cerebellum needs a lot of information and ways to adjust muscle action. Gross anatomically, the cerebellum doesn't look too complicated. Huge amounts of gray matter on the outside, and there's a little bit of white matter called arbor vitae. This doesn't say that cerebellum would be anything special. On the other hand, when you look at the connections of the cerebellum through the cerebellar peduncles, here is the secret of the cerebellum. So we have incoming sensor information through the spinal cord. Proprioception, just to say a few words about it before chapter 16, proprioception is the sensory information that tells our brain how our body is acting or positioned in three-dimensional space. muscle spindles, Golgi tendon organs that you saw in chapter 13 are proprioceptors, proprioceptive sensory structures. We have additional structures, some in our skin, some in our joint capsules, and so our cerebellum knows exactly which muscle is contracted to what degree, what is the angle your elbow or knee or hip joint is bent, and just how much load is on each tendon in your body. All that information is streaming into the cerebellum through the inferior cerebellar peduncles. What the brain wants to do, motor instruction that goes to skeletal muscles is split from the pons, and so the cerebellum knows what our consciously working part of the brain wants to do, like play piano or ride a bicycle. And the superior peduncle carries correcting nerve impulses back to the cerebrum, to the motor control areas, and we'll in part 2 of this chapter 14 we'll talk about these areas, name them by structure. So the cerebellum gets a lot of information through the inferior middle cerebellar peduncles, and it can change how our muscles are activated by influencing the cerebrum. And the last part here that we talk about is diencephalon. This is probably the least well-known portion of the brain. We see some very large structures like the thalamus, and a lot smaller structure like hypothalamus, and we have the third ventricle here as well. So this is a somewhat small portion of the brain, and you probably haven't heard a whole lot about diencephalon. On the other hand, this portion here, the hypothalamus, is so essential to maintaining homeostasis that it's worth spending time here with the diencephalon. So let's talk about the thalamus first. In AMP2, we'll come back to this as a major switching point of sensory impulses. So any nerve impulses coming from our retina get switched to a new neuron here. Nerve impulses coming from our taste receptors, from our hearing receptors, from our touch, pain, heat receptors are all getting switched to a new neuron. So thalamus is big. It's gray matter loaded with synapses that are switching material onto new neurons here. So it's a relay center in the diencephalon. The hypothalamus is something out of this world. On this artistic rendition, it looks like a bunch of jelly beans just stuck together here. We have several reflex centers here. All of these functions that we'll be mentioning here are hardwired into the hypothalamus, into this part of our nervous system. And so many automated reaction to imbalance of homeostasis gets readjusted here. And so let's say a few words about the hypothalamus. So adjusting homeostasis is a major role for organ systems in our body, right? This is a chapter 1 material. Hypothalamus is well informed. It receives a lot of nerve impulses from our skin, taste, smell, hearing, and also visual receptors in the body, our retina. The hypothalamus has several chemical labs running in it, measuring ion concentration, osmotic pressure. It measures hormone levels, measures temperature of the blood, it is active in many, many ways. So let's see a few examples, more specific examples. So let's start at the bottom here. Regulates states of consciousness. What the heck does this mean? Well, this means states of consciousness means wakeful state versus sleep. Circadian rhythm is the natural rhythm of the body. We become sleepy when it gets dark outside, and we are awakened from our sleep when the sun is coming up. That visual input into the hypothalamus programs the hypothalamus to send nerve impulses to the activating center, the reticular activating system that will wake us up in the morning. And when the hypothalamus stops sending nerve impulses to the reticular activating system, then we fall asleep. So this is the hypothalamus is the instigator of sleepiness, waking up, and if you ever had jet lag, if you travel way out of this time zone, then our hypothalamus becomes confused that the sun is rising early or sun is setting earlier or later, and jet lag is the perception, what we feel when our circadian rhythm from hypothalamus doesn't match actual reality around us. It takes 4 to 6 days to adjust to it. Our hypothalamus gets reprogrammed by the visual input. Let's talk about control of body temperature. You saw the eccrine sweat glands in our skin. How do they get turned on when we are overheating? Well, the hypothalamus receives nerve impulses from heat receptors in the skin, but the hypothalamus also by itself measuring the temperature of the blood flowing through it. When the hypothalamus detects hyperthermia, it sends nerve impulses to all the eccrine sweat glands and sweating starts. So controlling body temperature, and that's just one of the effect of the hypothalamus. So controlling body temperature can be done by the hypothalamus. In the chapter 27 of HAPch14, we will look at water balance, how we maintain water balance in our body, and drinking is one way to replace missing fluids. Guess what tells us it's time to drink, makes us thirsty? It's the hypothalamus that coordinates all kinds of nerve impulse signals from the rest of the body that tells us we are getting dehydrated, replenish fluids. Super important life-saving function, just this. Forget about the others. And there is a ton more. Hypothalamus, and this is more realm of the psychology, regulates emotional and behavioral patterns. It can moderate large swings in behavior when it's working normally. On the top here, this is going to come back in chapter 15, autonomic nervous system. This automated responses to changes in the environment by coordinating many organ systems at the same time. This is set by our hypothalamus. So whether we are in a relaxed rest and digest state or we are in a fight or flight response mode, our hypothalamus decides what we are doing actually. And the last one, this is also HAPch14 topic, produces hormones. And you have to say, gulp, what? This is not an endocrine gland. Well, many organs in our body create hormones, and hypothalamus is one of them. It's unusual because you wouldn't expect nervous system to create hormones, but hypothalamus does make antidiuretic hormone. Again, very important for resisting dehydration. And hypothalamus also makes oxytocin. Vaginal birth would not be possible without oxytocin. So hypothalamus saves our butt a number of times, and a lot of details will, a lot of additional details will be coming in AMP2. Let's name this portion of the diencephalon the epithalamus. This is a tiny posterior corner. The pineal gland is an endocrine gland. It creates melatonin hormone, and it is adjusting the circadian rhythm and coordinates its action with the hypothalamus. And lastly, let's say a few words about a group of organs that are missing the blood-brain barrier. These are the circumventricular organs. They are around the third ventricle. Why would the hypothalamus be missing? At least some parts of the hypothalamus would not have blood-brain barrier. Think about when you are creating hormones and secreting hormones. You don't want to secrete hormones from the other side of this blood-brain barrier. Measuring temperature is also easier that way. The pineal gland, again, creates hormones. Releasing hormones into the bloodstream would be hindered by the blood-brain barrier here too. And same is true for the pituitary gland. This is a major hormone producer, endocrine gland, and endocrine glands would be hindered in their activity by having blood-brain barrier. So this is the end to the first part of chapter 14, the brain. A lot more fun to come our way in the second part.